All right. First question. So here is what we call a serological test in which the positive result exhibits clumping. This is known as what type of test? Antigen test, anti-sera or anti-sera, agglutination, or ELISA.
You want me to answer it right now?
Yeah, just answer it on here. You don't need to say it out loud. All right, should we take a look? All right, this was actually better than the morning class. Okay, so about 2/3 on the right one. So, and actually, this breakup is very similar to how the morning class was too. So it's agglutination, and agglutination basically means clumping. Anybody heard of hemagglutination tests? Or hemagglutination where red blood cells clump? Usually it's a sign of flu. In these tests here, what we have are antibodies that will bind to an antigen, and I'm going to bet that's where antigen came into play. So antigen is just a piece of, in this case, a bacterial pathogen. And what we have are bacterial cells. And oftentimes this is used to, for example, distinguish different strains of, or different substrains, subspecies of E. coli from each other. So O157 versus O26. And again, that O refers to the core part of the or to the O part of the polysaccharide. And what we have are then antibodies will bind to multiple cells at one time, and it creates almost like a lattice-like network. And those antibodies are specific to one of those O polysaccharides. This is a pretty quick test. 10 minutes is about what it takes to see this clumping. And I do know these work because apparently folks have already been approaching them today, because I mentioned this earlier. There's somebody on campus in bio who's sick with mono. If you were in lab at 10 o'clock last week on Friday, we came in looking for a mono agglutination test, and it worked out. It was wonderful, and we found out he had mono. So, and for that one, we just used blood. Anyway, so this is an example where on the left is a positive test. That's the clumping. Right-hand side is a negative test for this. In general, there is a group of tests that are known as serological tests. All of them use this antibody method with either whole blood or plasma as a way to determine if there's some sort of reaction taking place. Okay. Any questions on this one? All right. All right, next one. Measuring the GC content in bacteria classifies bacteria based on their genome, phospholipids, proteins, or morphology. I already hear a question being asked. What does GC actually mean? It's a great question. That's important to figuring out the answer. So as you guys are plugging in the answers, I'm not grading these by any means, but where do we see G's and C's together?
DNA.
Nucleotides, right? GC is base pair. So in this case, it's genome. It's basically you're measuring or classifying the genome. We can calculate percentages either by looking at like melting temperature of the DNA or just by sequencing it. And through these methods can help to at least classify bacteria to the species level typically. Not much more resolution than that. A good method, not a perfect method, and most of that is true. That most of these will allow us at least resolution to the species level. We won't even look at how we did on that one.
No, come on.
No, I don't want to. All right. Do you guys want to? Because we can. All right. Oh, 83%. You guys did great.
You had no faith in us, Zach.
No, it was just because the questions started firing. What the hell does GC mean? What do you think I'm going to guess? All right. Here's the last one. 16S rDNA encodes cytoplasmic protein, subunit of the ribosome, tRNA, membrane protein, or peptidoglycan. Just rolls off the tongue. What? That's
the question.
oh.
See, because it gives you.
What's R stand for here? It encodes for an rRNA, which encodes for a part of the ribosome. Okay, so this one, the correct answer would be a subunit of the ribosome. I bring this one up for a couple of reasons. One, about the 1970s, there became this big way to change how we classified organisms. It's based around this molecule right here. So in eukaryotes, we look at the 18S. In prokaryotes, we look at the 16S. This is the actual RNA itself. And what I want to point out here are a couple of features. Anything that's not shaded, highly conserved, regardless of what species of bacteria you're talking about. The areas that are shaded are known as variable regions. Those are typically what gets sequenced. And by looking at those variable regions, we can determine, at least to the genus, sometimes to the species level, the identity of a bacteria. It's not perfect. If you're trying to determine substrains or subspecies, for example, you have to do more sequencing. You've got to actually sequence additional genes as well. The other thing about this is that and the reason why these regions are variable, or at least thought to be, is that it leads to slight changes in structure. Those slight changes in structure are not enough to affect the function of the ribosome. And it's been assumed that these mutations occur at some relative consistent rate. So we can use that, compare the sequence of two bacteria with each other in the 16S region, and determine not only how closely related are they, but when was their last common ancestor, just by making predictions about how frequently do these mutations occur. Okay. The reason why I point this out is we are going to talk about we're going to talk about human microbiome, and we'll talk about more environmental microbiomes later on in the semester. This is the technique that is oftentimes used to sequence, on the order of 1 million to 2 million bacterial cells in a 24-hour period. And you can classify them then, at least to the family level, as being part of the enterics, in other words, the ones that live in your gut, versus the Staphylococci family, or the Bacteroides. So this is something we'll come back to later on. So what I wanted to highlight with this is these are two of the ways that, I guess I'll go to the answer because you guys want to see that. I'm going to, sure, huh? How many people answered afterwards? Nobody's going to fess up. Okay. All right. There we go. All right. So what I'm trying to point out with those is there are 3 ways that we talk about in here that we can classify bacteria or identify bacteria. One is with anti-sera tests or serological tests. The second is by sequencing, looking at DNA. And then the third is with using biochemical tests, or we might call them metabolic tests, phenotypic tests, as a way to determine what actually are these genes expressing and how do they respond to the environment. And those are the ones that we most often will use in here, are those phenotypic tests. So for example, Wednesday you gram stained your unknown, you determine not only gram status but morphology. Okay? And then in about a week and a half from now, you'll begin to do some of the metabolic tests as well to determine right, so the one that is the one that has two sides and are the two tables. Focus first on the side that's just P. aeruginosa, 3 different E. coli, Salmonella, Shigella, Pasteurella, Klebsiella. That one's just like the activity we just did, though it's testing for some different things. So for example, the O26 antibody is a serological test. The STX gene is actually sequencing DNA test. That one this one is just a practice review for yourself. It's create a key the same way that you just did. If you're feeling good with that, I encourage you to try the last one. So the other side of that sheet, the other table, correlates to this bigger one here, where I've got the names listed, but the questions and whether it's positive or negative aren't included. This is one just kind of like a puzzle. Work backwards to figure out what are the actual questions, or what questions could you ask to distinguish them from each other. Okay? I'm not asking you to hand it in, but say you get bored because the Bears are doing really bad at 12 o'clock on Saturday or Sunday, and you're just like, hey, I got a dichotomous key I could enjoy for a while. Pull out the dichotomous key. Try it out. Okay? And I will look at these if you want to, but do not feel obligated to hand them in. Danica asked, will this be on the test? Absolutely, there's going to be a dichotomous key on the test. Either you're going to have to draw one, interpret one of the two. Okay, there's one every single semester. There's a dichotomous key question on the exam. And it falls usually under the essay category, even though you're not writing anything other than questions. As for the one that I kind of mentioned or alluded to as an assignment, so by Lab 8, which unfortunately for you guys falls on the same day as the exam, sorry. Dichotomous key for you and your partner, you create one. It's a 20-point assignment, but it's basically in the lab manual. And I think I've got this posted up online too. If not, I will post it. It's a large Excel spreadsheet of all the different tests that you have available to you in lab that you can use to determine is your or what is the identity of your unknown. And you and your partner will just hand in to me a 8.5 by 11 sheet of paper, or 11 by 14, whatever you prefer, handwritten, printed out, of that dichotomous key. And that's what you'll use to help you identify your unknown. So that's why I have you hand them in ahead of time for when we do that. So just to kind of give you a heads up that that's coming, it really does not, I don't think that it takes too long, but I also have not done it myself in many years. So, okay. Couple of last things for today. We'll get out of here early otherwise. Next week we move into metabolism. And who does not like talking about metabolism in bio? Raise hands. It's fine with me if you don't like it. Thanks, Ed. Yeah. You just did that for the sake of it.
Did we talk about metabolism?
Not much. Why do people hate metabolism so much? Typically because it's a lot of memorization, right? Who's had to memorize glycolysis or steps like that? I know Matt has, right? Probably biochemistry. Okay, here's my thought on that. It's a waste of time. I find the memorization to be a complete waste of time because you probably will never have to do it again in your life. Okay? So, the way that I like to go through metabolism is rather than think about it on like having you remember every single enzyme, every single substrate and product in glycolysis, or all the different molecules that show up in Krebs cycle. Instead, what we're going to do is we're just going to use those as examples of metabolic pathways, and we're going to talk about this more on a global scale, how they're mechanistically linked to each other, how they're affected when one of them may get shut down. And then instead of focusing on specific enzymes, we're going to talk about classes of enzymes. So for example, there is one step in glycolysis. Fructose 6-phosphate goes to fructose 1,6-bisphosphate, and then there's an enzyme in there known as phosphofructokinase. What I want you to be able to do is identify, hey, there's a phosphate involved, and it's going from a substrate to a product. That means there must be a kinase enzyme involved. So we're going to talk more in terms of general themes that you would see rather than the specific enzymes themselves. Okay? And then there's going to be focus on how does this correlate then to these metabolic tests in the lab. So that's the first heads up. But for Monday, everybody's favorite type of assignment,
Yes.
voice thread. So, what are we going to do? Everybody right now assign one member of a group to pull out their computer and set up the group voice thread. No, actually, Sid, I wanted Sid to do it because you're the one who has the most trouble. All right, we're going to walk through this together, too, to make sure that it gets set up.
No, don't do it. Okay, if I make it, you gotta post it. Because I make it, you gotta post it.
All right. It should be, so you should have a new tab in the assignments that's microbial microbial metabolism lesson 1. It's under the assignments. Make sure you're using Chrome or something like that. All right. I'll keep waiting. I'll give you like 1 more minute.
I got it.
Don't leave me behind. It's not good.
You want us to watch the video?
No, don't right now.
Should we just create one?
Create one. That's actually the first step. Hit create.
It says add media.
Oh, I need to put this up, sorry.
I don't have a media.
Yeah, I don't have a media to add.
Oh, no, that's okay. So I'm going to show you how to do this. Ready? If you don't have a media to add right now, do this. Click on the plus button. Take a picture.
Oh, sweet. Okay.
No.
And it's just going to upload it. See? Right
I got a bootleg.
there.
You gotta make it.
And then give it a title. No. You could just do a quick voice if you want to, like test 1, 2, or something like that. All right, it's Friday. There's my new voice thread. All right, we good with that? Next step, share. Click on share. What? Whatever you want to. Enzymes. Most awesomest group. If you want to stop calling yourself by numbers and start calling yourself something else, that's totally cool. All right. So the share button is right here. Why don't you?
It's going as group 17 again.
Damn it! That's not the only one.
Wait.
Okay, who does not have the option to actually share this?
I can't share it with people.
I can share it.
I can allow people to comment on it.
Okay, we need to do more than that. Okay, I will help those of you that are having trouble sharing in a minute, okay? Because there's a group of them. But no! You hit share. I'm going to show you how to share once you actually can. Okay?
Wait, why can I do it though?
I don't know. Click on numbers.